Hello everyone, grab your pen and paper and let's talk about primary immune deficiencies. In this lecture, we're going to be going through an overview of what primary immune deficiencies are and what they mean, and we're also going to be talking about a few examples to help us illustrate what we mean. First of all, we know that lymphoid stem cells can give rise to both B cells and T cells, and if there were problems related to any of them or both, they're going to be having a negative effect on our body. First of all, let's talk about both B and T cell deficiencies. Now, in some rare cases, our bodies will have an adenosine deaminase deficiency. This is due to the absence of this enzyme, and this is called ADA, and it is going to be causing severe combined immunodeficiencies. Now, in here, we have to just focus on the point that when we mean combined, we talk about B and T cells. So essentially, since we do not have this enzyme that is going to be breaking adenosine, that means that this adenosine is going to be accumulated in our body. Now, this is going to be toxic, and this is going to be killing, well, both our B cells and our T cells. The resultant from this is the lack of virtually all immune protection, since we know that T and B cells comprise the majority of our efficient immune protection, and this is going to be the only immune protection essentially that we have is the innate one, and it is not as strong. Let's start with some B cell examples when we have a deficiency in B cells. The first example is Bruton's A gamma globulinemia, and it is characterized by the loss of the Bruton's tyrosine kinase, or the BTK gene, and this is going to be leading in complications related to the signaling that is going to be producing mature B cells, essentially plasma cells. Now, as we all know, plasma cells are going to be giving us antibodies, so if we have a problem related to the formation or the production of these mature B cells, or plasma cells in this case, then we're not going to be having any antibodies, or the antibodies are going to be in very few numbers. So some symptoms include low levels or the complete absence of B cells, which causes patients to be susceptible to recurrent infections, since we also know that B cells or plasma cells are going to be producing memory cells or memory B cells, and these are going to be very important to protect against recurrent infection. Now, worth mentioning that the BTK gene is going to be present on the long arm of the X chromosome in the position XQ21.22. Now, the second example is the selective IGA deficiency. Now, it is very direct. The title says it all, essentially. We have a lack of IgA antibody specifically that is present in our body. Now, the main function of the IgA is to protect against infections in the mucous membranes lining the mouth, the airways, and the digestive tract. So naturally, if we have a problem with this IgA antibody, then we're going to be having more complications in these areas. So IgA deficiencies, while significant, can go unnoticed, essentially asymptomatic for years. Now, this can be triggered. We're going to be knowing that there's a lack of IgAs when we have a blood transfusion, because, I mean, obviously, if we want to get a blood transfusion, IgAs and all antibodies are going to come with the blood of the donor. So if our body receives any IgAs, or any antibody for that matter, but specifically in this example, the IgA, what's going to be happening is that since it does not know, it is not considered as self antibodies, our immune system is going to be triggering an immune response against this donor's IgA, which is going to be leading to complications. This is when we actually have, or when we know that we have a deficiency in this antibody specifically. Now, that being said, patients suffering from IgA deficiencies are more susceptible to pneumonia, just like we said before, and episodes of respiratory infections, as well as autoimmune diseases. Next, we're going to be talking about T cell examples. So some T cell examples, we've got the DiGeorge syndrome, and it occurs when patients lack parts or completely lack the thymus. Now, as we also know that the thymus is the location where the T cells are going to be maturing. So there's going to be a problem in the thymus due to embryonic development problems, absence of the third and the fourth pharyngeal pouches in the embryonic stage. Now, in addition to their lack of thymus, they also might lack the parathyroid glands, and this is ultimately going to be leading to a lack in the release of parathyroid hormones, which is also going to be leading to further complications. Now, it's worth noting that T cells mature in the thymus, and therefore its absence will result in the inability of T cells to mature. This syndrome develops due to the deletion of the 22q11.2 region in the chromosome. Now, the consequences of this syndrome include the increased susceptibility to intracellular parasites like pathogens and intracellular bacteria. Let's try to explain why that is, because since we know that T cells, generally speaking, are going to be they're antigen-presenting cells, so they're going to be they're going to be killing pathogens. They're going to be taking a part of their proteins or their sequence of their amino acids, and they're going to be presenting them for other cells to trigger an immune response. Essentially, this is what happens. Now, if we have a lack of these cells, what is going to be happening is that there is no immune response that is going to be rendered against those pathogens. That is why these infections are more recurrent. Thank you for watching. This has been Ali from B Biology.